Hi guys, so it's Madison and I'm going to do my virtual poster presentation in the form of a PowerPoint based on my project that I've been working on. So I've been working in Dr. Lowry's lab through the Department of Integrative Physiology for a while now. So he studies behavioral neuroendocrinology. And my project that I helped my mentor Christian with was titled Mycobacterium vaccae modulates brain circuits in response to stress in Sprague Dawley rats. So the goal of my project was to identify brain regions known to affect stress and stress reactivity in response to Mycobacterium vaccae treatment. So essentially, we're trying to figure out how and where this vaccae bacteria is working in rats. So our hypothesis for this project was that the vaccae will reduce the activation of the CRF neurons, and we would see a decrease in these double-stained neurons in our animals that were treated with vaccae, because that means that their response to stress was somehow mitigated by the bacteria. So why vaccae? Why is this random bacteria so important? So this goes back to the old friends hypothesis, which this is saying that the sterile environment that we live in today in modern life has reduced exposure to beneficial old friends, so beneficial bacteria. And this decreased exposure has thrown off the balance of beneficial bacteria that exist within our gut microbiome. And why the Lowry Lab studies vaccae is because it has been seen to incur anti-inflammatory and immunoregulatory properties, which are conferring these stress-resilient properties in rats. And this may prevent stress and trauma-related psychiatric disorders in humans, such as depression, PTSD, and other inflammatory diseases such as those. So as far as the methods go for my project, the rats were immunized with the vaccae bacteria once a week for 3 weeks, leading up to day 1 of the experiment, when they were exposed to a stressor to kind of induce the effects of vaccae, and this came in the form of an inescapable tail shock. And then on that day 1, the animals were sacrificed. After that, they were perfused with PFA and their brains were frozen. We then sliced up their brains, and after that, we stained their brains with antibodies using immunohistochemistry, and then took pictures, and we've been working on counting, so quantifying the areas of activation that we saw of staining in these brains. Okay, so we used c-Fos and CRF to stain our samples, and this was showing where activation was happening from this vaccae. So c-Fos showed up blue on the samples and CRF showed up brown. And when we saw cells that were brown with blue nuclei inside, kind of like this one in this picture right here on the right, we could infer that there was activation to the point where a response was induced in this cell to the stimuli of the stressor that the rat underwent before it was sacrificed. And we were looking at these regions within the BNST and the CEA, which I'll talk about later, in order to quantify the amount of activation that was occurring in these areas. And this isn't a picture from my experiment. Just to mention, this is an old experiment that Dr. Lowry participated in because my presentation or my project isn't quite finished yet, and the results and conclusions and everything haven't been published. I just used this old picture so that way you could see kind of what I was looking at when I was looking under the microscope and counting and quantifying the activation in these areas. Okay, so where did we look in our animals? So as I mentioned before, we were studying the BNST and the CEA regions of the brain, and these are related to stress and fear responses in the animal. And within those regions, we looked at several bregma levels, which means we looked at different layers within the brain to give a more comprehensive look at those areas that we chose. So we have lots of problems with this staining. Immunohistochemistry is tricky, to say the least. So first of all, we were just out of the c-Fos antibody that we normally use in the lab, and we needed to replace it with pretty much like a novel strain of this antibody. We had lots of optimizations, meaning we had to do lots of tests to see if the new antibody that we would order was working. That took a while. And then non-specific binding of antibodies caused a lot of background staining. So basically, whenever you're looking at under the microscope, it was clouding what we were trying to look at. So those are a lot of problems that we experienced with our protocol. So that kind of set us behind. So basically the project is still a work in progress. You can see that I was fortunate enough to get permission from the lab to bring my microscope home for a couple days to work on quantifying. So that was pretty cool. So as far as next steps go, we need to finish quantifying, and then based on that we can make statistics and figures that will help us to draw conclusions about whether or not vaccae is working in the BNST or the CEA regions of the brain, and then draw conclusions about the bacteria based on that. And if we don't see anything in those areas, then we just will look to figure out what region we can look at next. And I just wanted to thank Dr. Lowry, my PI, Christian Zambrano, postdoc that has been my mentor throughout this whole process, and then Eduardo Primini, who's a PhD student who has been so willing to help both Christian and I with some of the more tedious parts of the project. It was really great of him to step in and help us. So that's all I got for you guys. All right, thanks.